This presentation is to help you to understand the basics of interpreting a disease pedigree. A pedigree is, of course, a chart showing the ancestry of a person of interest. We call this person the proband. The pedigree, if prepared and interpreted properly, can give us insight into the genetic nature of a disease. There are two questions you have to ask when approaching a pedigree. First, are there carriers? In a disease which is inherited through a dominant gene, by definition, there can't be any carriers. If you get one copy of the gene, you're toast. If the disease is recessive, then people who have one copy of the gene are called carriers, and people who have two copies of the gene are toast. Second, are the sexes represented equally? A genetic disease results from a mutation on one of the chromosomes 1 through 22. These are called autosomes, or on the X or Y chromosome, sex chromosomes. If there are only female carriers and only males are affected, then we know the disease is X-linked recessive, which we also call sex-linked recessive. Here's why. We can simplify our lives by remembering that very, very few diseases are sex-linked dominant, that is, caused by a single mutation on a single X chromosome. Even fewer diseases result from mutations of the Y chromosome, so let's say none for our purposes. So if sex-linked genetic diseases are almost always carried on the X chromosome and almost always recessive, then females are carriers for these diseases. They have one good X chromosome and one bad. Males, on the other hand, can't be carriers because they have a bad X chromosome and a Y chromosome that doesn't help one way or the other, so they're always affected by X-linked or sex-linked diseases. In a pedigree, males are represented by squares, females by circles. Affected individuals are represented by filled symbols, carriers by half-filled symbols, and unaffected individuals by open symbols. This first pedigree is autosomal dominant. There are no carriers, so we know it's dominant. Both males and females are affected, so we know it's autosomal. If you plot the inheritance out as a Punnett square with the mother as big D little d along the side and the father as little d along the top, we see that 2 out of 4 children on average will be affected and 2 out of 4 children on average will be unaffected. In other words, each child has a 50% chance of being affected. This second pedigree is autosomal recessive. There are carriers, that tells us it's recessive. The sexes are affected equally, and we see both male carriers, not postal workers, but MALE carriers, and female carriers. The Punnett square at left gives us the proportions. In a mating of 2 carriers, as shown in the middle row of this pedigree, 1 out of 4 of the children will be affected, inheriting both bad copies of the gene. That's 25% chance of inheriting both bad copies. That's shown in the lower right-hand corner of the Punnett square and the far left symbol on the last row of the pedigree. 1 out of 4, or 25% of the children, will be clear, inheriting both good copies of the genes. That's shown in the upper left corner of the Punnett square and the clear symbol second from the left on the bottom row of the pedigree. And 2 out of 4 children will be carriers, inheriting 1 bad copy of the gene and 1 good copy of the gene. These are the half-filled symbols at right on the pedigree and the last row of the pedigree, and at upper right and lower left on the Punnett square. The last pedigree we're looking at is X-linked recessive. The females are carriers, shown by half-filled symbols in the pedigree, and XX individuals in the bottom row of the Punnett square. Males are either affected if they inherited a bad copy of the gene on the X chromosome, as shown by the second symbol from the left in the pedigree and at the top right corner of the Punnett square, or unaffected if they inherited a good copy of the gene on the X chromosome, as shown by the far right symbol on the pedigree chart and the upper left corner of the Punnett square. If that's all you want to know about pedigrees, and it's all you need to know for the exam, then you can stop the presentation here. If you're curious about how pedigrees look in the real world, I've got some examples from the scientific literature to show you. You don't need to know these, but I think they'll explain how we use pedigrees to better understand human disease. This pedigree is from a 1967 paper by Gilardi and Brown. The ages of the probands are shown as numbers, and if they're deceased, there's a small d next to their ages. A number inside a symbol indicates how many of that gender are born. So, for example, the 78-year-old female at the top of this pedigree had 4 sisters. We suspect, but don't know for certain, that she had the disease, so we give her a stippled symbol instead of a filled or open one. Sometimes we don't know or don't care what the gender is, so we give these people a triangle, like Tinky Winky here. The authors call this disease autosomal dominant. Can you see why? There are no carriers. There's a 50-50 chance on average of inheriting the disease, and the disease is represented in both sexes. Here's an example of a pedigree from a classic paper on Christmas disease, named that because it was first identified in a family with the surname Christmas, and also because the paper was published Christmas week in 1952 in the British Medical Journal. No carriers are shown not because they don't exist, but because we didn't have genetic testing in those days, so we had to guess at who the carriers are. Only males are affected. Females are unaffected. All 3 of the females in the second row are carriers. So if we have female carriers and only males are affected, then what kind of disease is this? It's sex-linked or X-linked recessive. The disease is now called hemophilia B and is a relatively rare form of hemophilia. It's caused by a factor IX deficiency. In this pedigree, we see the problem with something called consanguinity, marriages between first cousins. In this pedigree, deceased individuals have a line through their symbols. 2 cousins have married, and 2 of their children, both female, were born without eyes, a condition called anophthalmos. This is obviously a heartbreaking outcome, and we can see why first cousin marriages are outlawed in many societies. This pedigree is tough to figure out. Transmission seems to be through the females in this pedigree, but it's not X-linked. The authors call this pedigree non-Mendelian maternal transmission, which means it's neither dominant or recessive, but some other kind of genetic transmission. How that works is beyond the scope of this course, but you should know that not all genetics is simple Mendelian. We'll see lots of examples of non-Mendelian genetics in the clinic. What in the world is going on here? This is one messed up pedigree. One female has mated with her uncle, and they had 18 offspring. A male who's labeled as a carrier has mated with both his aunt and great-aunt. If we look at the title of the paper this came from, we get a clue. This is a dog pedigree. Still, the same rules apply. Carriers are represented by half-filled symbols. There are both male and female carriers,